Assalamu alaikum doctors, welcome to my YouTube channel MBBS and BDS lectures. Today is the sixth video of rapid revision course of Periodontics, which is very important for NLE exam, MDS as well as for BDS students. In this video we will discuss about Cementum in detail, we will cover everything about Cementum. First we start from how we define Cementum, then who demonstrates the Cementum, and Cementum cells, then Cementum chemical composition, then Cementum classification, and then at the end the abnormalities in Cementum structure. First we start from definition. Cementum is a calcified avascular mesenchymal tissue. Remember, cementum is avascular calcified mesenchymal tissue that form the outer covering of the anatomic root. First demonstrated microscopically in 1835 by two pupils of Purkinje. Formed throughout life. Cementum formed throughout life. Cementoblasts are the cementum forming cell, and when it mature it become cementocyte. Cementoblasts, which are initially separated from cementum by uncalcified matrix which is known as cementoid, sometimes become enclosed within a matrix and are trapped. When it is enclosed within a matrix and are entrapped, once they are enclosed, they are referred as cementocyte. Cementocyte are the mature cementum cell. Look at below diagram. When they enclose in the cementoid matrix, then it is called cementocyte. Now come to the chemical composition of cementum. Remember, the inorganic portion of cementum consists of calcium and phosphate, which form the hydroxyapatite crystal. Now, cementum is less mineralized than bone, enamel, and dentin. Remember, the inorganic portion of enamel is more mineralized than all of tissue in our body. Like enamel have 97% inorganic content, then dentin have 70%, then bone have 65%, and cementum have less amount of inorganic crystal, mean calcium and phosphate, to form hydroxyapatite crystal, which is only 45%. Now, how you remember chemical composition of cementum? It is very easy. Remember by 50-50 concept. In cementum, 50% is inorganic and 50% is organic. And in 50% minus 5, there is 45% inorganic and 55% is the remaining part. Now here in table, the by volume it is very important. Inorganic material by volume is 45 and organic material by volume is 33 and water is 22. By weight, inorganic is 65, organic is 23 and water is 12%. On dry weight basis, inorganic is from 45 to 50 and organic is from 50 to 55. Mean simple. Inorganic is content is from 45 to 50 and organic from 50 to 55. This is a chemical composition which are very important for MCQ's part. Now come to the organic matrix. The major portion of organic matrix of cementum is composed of type 1 and type 3 collagen fiber, but type 1 is 90% and type 3 is 5%. Next is the fiber, mean organic in organic part there is the collagen fiber, which are of two types. Mean from two sources, one from outer source, which are extrinsic fibers, and one from inner source, which is intrinsic fiber. Now the outer source extrinsic fiber is the sharpest fiber, which is actually the principal collagen fibers of PDL, which are enter in cementum, and next is the intrinsic fiber, which are cementum own fibers. Now, Sharpie fibers, which is extrinsic fibers, and fibers that belong to cementum matrix, these are intrinsic fibers and are produced by cementoblast. But Sharpie fibers is produced by fibroblast of periodontal ligament. Now, other is the cell types, and next in organic component there is a different types of protein like bone shallow protein and osteopontin. Next is the cementum attachment protein, and next is the cementum derived growth factor. These are the organic part. Mean in inorganic part there is a calcium and phosphate which form hydroxyapatite crystal, and in organic there is a type 1 and type 2 collagen fiber, which source is extrinsic and intrinsic. Next is the other cell types, then the different types of protein like bone shallow protein and osteopontin, cementum attachment protein, and cementum derived growth factor. Now after chemical composition, now come to the classification of cementum. First, cementum is classified according to cellularity and time of formation. According to cellularity, either cell is present or absent, and time of formation. Either cementum formed before tooth come in the occlusion or after tooth come in the occlusion. On the basis of cellularity and time of formation, we divide cementum into two types. Number one is the acellular cementums or primary cementums, which have no cell. Other is the cellular cementums or secondary cementum, which have cell or cellular cementum. Now first, acellular cementums. The acellular cementum is formed when tooth are not come into the occlusion. The acellular cementum is formed. It is the first cementum found. The first cementum which are formed is the acellular cementum, cover approximately cervical 1/3 or half of the root. Mean it is present in the cervical 1/3 or half of the root, and its thickness is from 30 to 230 micrometer. Next is the cellular or secondary cementum. It is formed after the tooth reaches the occlusal plane and it is more irregular and contains cementocyte in individual spaces, which is known as lacunae, that communicate with each other through a system of anastomosis canali coli like in bone. Now here the next important is the what is incremental lines of Salter. These lines represent rest period in cementum formation and are more mineralized than adjacent cementum. Mean between primary and secondary system cementum formation, this is the rest period which is known as incremental line of Salter, and they are more mineralized than adjacent cementum. Now come to this very important table, which are very important for MCQ's point of view. There are the 10 differences between acellular and cellular cementum. First, formation. Acellular cementum form before tooth reach occlusal plane, and cellular cementum form after tooth reach occlusal plane. Next, cell. In acellular cementum do not contain any cells. Cellular cementum contain cementocyte, which are the mature cementum cell. Third is the location. Acellular cementum are present on the coronal portion of the root, and cellular cementum present on the apical portion of the root. Next is the rate of formation. The rate of formation of acellular cementum is slower and cellular cementum is faster. Next is the incremental line. In acellular cementum, incremental line are more, and in cellular cementum, spare mean less. And next is the in acellular cementum, function form after regenerative periodontal surgical procedure. Mean its function is formation after regenerative periodontal surgical procedure. When we do regenerative periodontal surgical procedure, when we do it form, its function is formation. Next is cellular cementum contribute to the length of root during growth. Mean it form the length of the root because it are present on the apical portion. Next is the calcification. Acellular cementum are more calcified, cellular cementum are less calcified. Next is the Sharpie's fiber. Sharpie's fiber are the extrinsic fiber of PDL which present in the cementum. In acellular cementum there is more Sharpie's fiber, in cellular cementum less. And next is the acellular cementum regularity. Acellular cementum is regular and cellular cementum is irregular. Next is the thickness. Thickness of acellular cementum is from 20 to 50 micrometer near the cervical region and 150 to 200 micrometer near the apex. In cellular cementum, thickness from 1 to several mm. There is a more thickened cementum. These are the differences between acellular and cellular cementum. Next, come to the second classification based on Schroeder classification of cementum. This classification is about whether cell is present or not, whether there is the extrinsic fiber or intrinsic fiber, or there is no cell, no fiber. On this basis, there are 5 types of cementum. Number 1 is the acellular afibrillar cementum. Mean AAC, there is no cell, there is no fiber. This is the acellular afibrillar cementum. These are present at the coronal portion. These are the coronal cementum and thickness is from 1 to 15 micrometers. Next is the acellular, mean no cell, extrinsic fiber cementum, but fiber is extrinsic, mean Sharpie's fiber from outside, not cementum own fiber. This present at the cervical third of the root in a human, but may extend further apically, mean it may extend apically. Thickness is between 30 to 230 micrometer. It form on the newly synthesized dentine surface. Next is the cellular mixed stratified cementum. Cellular means cellular present. Mixed means there are 2 types of fiber, both extrinsic and intrinsic. First is the acellular afibrillar. No cell, no fiber, means there is no intrinsic or no extrinsic fiber. Next is the acellular extrinsic fiber. Intrinsic fibers are not present. Extrinsic are present but no cell. In third is the cellular, means cell are present, and mixed stratified cementum. Mean mixed mean both extrinsic and intrinsic fibers are present. Primarily, it present in the apical third of the root and apices, and in the furcation areas. Thickness range from 100 to 1000 micrometer. Next is the cellular intrinsic fiber cementum. CIFC, cellular intrinsic. There is a cellular, but there is a not extrinsic fiber. There is an intrinsic fiber are present. It fill a resorption lacunae. It is commonly associated with the repair of the resorptive defect and healing of the root fracture. Mean during healing of the root fracture and in the to repair of the resorption defect, there is a cellular intrinsic fiber cementum formation occur. Last is the intermediate cementum. Intermediate cementum is a poorly defined zone near the cementodentinal junction. Now MCQs come where intermediate cementum is present near the cementodentinal junction, where cementum and dentin form junction. It is a poorly defined zone. It is also called layer of Hopewell-Smith. This is the Schroeder classification of cementum. Now come to the cemento-enamel junction. This is also important. Where cementum and enamel meet, this area is known as cemento-enamel junction. 3 types of relationship involving cementum may exist as the cemento-enamel junction. There are 3 types of cemento-enamel junction. Number 1 is the overlap, where cementum overlap the enamel. It is present in 60 to 65% of cases. Means in 60 to 65% of people overlap cementum is present. Next is the in 30% there is a edge to edge contact. Mean there is a butt joint formation, mean butt joint exists. There is a edge to edge contact. In 5 to 10%, there is a cementum and enamel fail to meet, and there is a depression in between cementum and enamel, and these people, there is the in this area the identity is exposed, and these people are more prone to dentin sensitivity. Now come to the thickness of cementum. The thickness of cementum on the coronal half of the root vary from 16 to 60 micrometer, or about a thickness of a hair, like human hair. It attains its greatest thickness up to 150 to 20 micrometer in the apical third and in the furcation area. Mean in coronal half there is less thickened, but in the furcation and apical 1/3 of root there is a more thickness of cementum. It is thicker in distal surface than in mesial surface. Remember, cementum is thicker in distal surface than in mesial surface. Why? Probably because of functional stimulation from mesial drift over time. Because to move in mesial direction, due to mesial drift, there is a functional stimulation. In the result, there will be more cementum on distal surface, because from mesial surface there is a stimulation come to the distal surface from the mesial drift. Next come to the abnormalities in the thickness of cementum. Abnormalities in the thickness of cementum is known as, first is the hypercementosis. There is a more cementum formation. The causes of hypercementosis are benign cementoblastoma, cementifying fibroma, periapical cemental dysplasia, fluoride cementoosseous dysplasia, other benign fibroosseous lesion. Now come to the radiographic feature. This is very important. In the radiographic feature, radiolucent shadow of the periodontal ligament and radio-opaque lamina dura are always seen on the outer border of the area of hypercementosis. Meaning, hypercementosis, the lamina dura are radio-opaque, and the periodontal ligament show a radiolucent shadow, and enveloping it is wood in normal cementum. Mean around the normal cementum there is a radio opaque lamina dura. The differential what is the lamina dura? And the lining of the alveolar socket. The lining of the alveolar socket are on the radiograph is called lamina dura. Now come to the differential diagnosis of the hypercementosis, the periapical cemental dysplasia. Next is the condensing osteitis. Again, in condensing osteitis there is an inflammation of the bone around apical margin, and there is a hardening of the bone, which are also radio opaque. And next is the differential diagnosis, there is a focal periapical osteoporosis. Next come to the changes in cementum after instrumentation. When we do instrumentation of cementum, what types of changes occur in cementum? When we do scaling in perio, and patient complain of hypersensitivity. Why? Firm scaling stroke used to remove subgingival calculus also remove a small amount of cementum. This is known as calculocementum, resulting in notching of the root surface, exposure of dentin, and subsequent hypersensitivity. Next is the developmental and acquired anomalies associated with the cementogenesis. Mean developmental and acquired anomalies associated with the cementogenesis. First are the developmental. There is the number one is the enamel projections. These are the enamel projections which are entered into the cementum. These are the extensions of the enamel. This is known as enamel projection. Next is the enamel pulse. This is also an enamel which present on the bifurcation area in the cementum. This is known as enamel pearl. And next is the cementicles. These are the stone which are formed in the cementum areas. Next is the cementum resorption. What is cementum resorption? The newly formed cementum, these are the acquired conditions. These are the acquired anomalies. And now, first we study the developmental anomalies, which is enamel projection, enamel pores, and symmetrical. Now come to this acquired anomalies, the deformities. These are the number one is the cementum resorption. The newly formed cementum is demarcated from the root by a deeply staining irregular line termed as a reversal line, which delineates the border of the previous resorption. Means cementum resorption is the acquired condition. In cementum resorption, the newly formed cementum is demarcated from the root by a reversal line. Next is the ankylosis. What is ankylosis? In ankylosis, the cementum of the root are fused with the bone and the PDL is destroyed. Mainly occur in trauma. In ankylosis, fusion of the cementum and alveolar bone with obliteration of the periodontal ligament. They have a specific metallic percussion sound. When we percuss the tooth, there is a special metallic percussion sound, and if ankylotic process continue, tooth will be in infraocclusion. How we diagnose the tooth? Tooth will be present in infraocclusion, mean below the other occlusal surface of the other teeth, because the PDL is damaged and tooth is directly fused with the bone. Next is the hypophosphatasia. Hypophosphatasia is the rare familial disease characterized by incomplete bone mineralization. This condition was first described by Rothman in 1984 and is characterized by low level of serum kidney and bone alkaline phosphatase and elevated level of phosphoethanolamine in serum and in urine, characterized by losing and premature loss of interior deciduous teeth. The exfoliated teeth are characterized by an almost total absence of cementum. This is all about cementum. In next video, we will discuss about alveolar bone in detail. And if you like my video, press the like button and share this video with your friends. If you are new to my channel, subscribe my channel by pressing the red subscribe button and also press the bell icon in order to receive my each new video notification. For watching my videos, I am very thankful to you. Thank you very much.